Good afternoon. Now I'd like to take the privilege to present before you the profile of our next speaker, Dr. M.V. Keat. Dr. M.V. Keat is an associate professor at the School of Data Sciences in Perdana University, Malaysia. She is currently acting the dean and head of the institution education unit at School of Data Sciences, Perdana University. She obtained her Bachelor of Sciences in Bioinformatics from the University of Malaysia in 2004. Before obtaining PhD in Structural Bioinformatics from University of Science, Malaysia in 2010. Dr. Yam started her journey in bioinformatics since undergraduate years and went on to specialize in structural bioinformatics during her PhD. Years lately, she has started venturing into education, research, and innovation with a special interest of reaching out the community about bioinformatics and data sciences. Over the years, she has obtained several research grants from the Malaysian government and university to conduct bioinformatics related works. She nurtures postgraduates and undergraduate students, research and interns throughout her career as an academic staff. Besides that, as a certified trainer herself, also actively trains and conducts trainings, workshops across Malaysia and Southeast Asian topics in bioinformatics, data science, and innovation and education. She is currently the Vice President, Education and Research for AP Bionet, Asia Pacific Bioinformatics Network. She is also the founding chairperson of My Bionet InfoNet, a virtual networking platform for Malaysian bioinformatics enthusiasts. She also serves as an EXO member for the Malaysian Society of Bioinformatics and Computational Biology. She was an affiliate member of the Young Scientist Network Academy of Malaysia and was a member of Royal Chemistry Society. Now I'd like to invite Dr. Ma'am M to deliver her talk on structural bioinformatics in drug discovery.
Thank you very
Ma'am,
much for the
please.
introduction. Just checking if you can hear me.
Yes, ma'am, you are audible.
Great.
Please go ahead. Yeah.
I'm going to share my screen now. So please also let me know if you can see my screen.
Yes, ma'am, it is.
Great. Thank you very much. So good afternoon, everybody, and thank you very much for this invitation. It's certainly my honor to be here. So as introduced by both the chairperson, you can see that I'm actually from Perdana University School of Data Sciences, and my topic today, I'll be having a quite a light topic because I thought maybe around this afternoon time you'll probably be prefer to have a light topic. So I actually decided to talk about structural bioinformatics in drug discovery. But I'm not going to talk more about the sciences, but more into whether is it still relevant today in today's context. Yep. So please allow me to share a little bit with you about where I'm from. I'm currently from Perdana University. As you see, this is a Malaysian map. So if we can zoom in, we are actually in Kuala Lumpur, very much downtown at Kuala Lumpur, the city center of Malaysia. So our building, our office building is currently at Damansara Heights, a very upbeat place in Kuala Lumpur, where we are connected with a lot of public transportation and easy access to many famous place in Kuala Lumpur. So this is how our campus look like. We are a one building block kind of campus with focus with students. And just to share some significant milestone for the university. The university actually have uh, two medical schools that we started in 2011. So we are relatively a new university. And at the same time, we also set up another school of foundation where we cater for pre-university student. And moving on, our school of data sciences actually was set up in 2015, and only last year we set up school of business. So you can see that the university is basically quite new and we are still in the midst of expanding. Yeah. So I'm going to move into talking about structural bioinformatics. I guess there are a number of people here who are probably interested in structural bioinformatics. But for the benefit of everyone, I'll probably just give you a very brief introduction about structural bioinformatics. This is basically a subdiscipline of bioinformatics whereby the main emphasis of this field is to focus on the representation, storage, retrieval, analysis, and display of structural information at atomic and also subcellular level. So whatever that we are looking at is actually very much into the atomic level. And of course, you can see that we have two main goals when we perform structural bioinformatics. The first is to create a general purpose to manipulate information about biological macromolecules. So we are basically focusing on the macromolecule. And another thing is to, of course, to apply enable to solve any problem in biology, and also ultimately is to create new knowledge. But there's always a very strong belief that in order to perform effective structural bioinformatics, there must be an availability of high resolution structural information about the biological system. And why do we need that? Because only high resolution information, high precision of information will enable us to get very good reasoning into the function and also the effects. So if we wanted to understand about a particular protein, about their function, about their how they perform their role in the particular system, it's very important for us to get very high quality input data. Next, I'm going to move into this perspective of looking into structure, because when we perform studies on protein, or even perform studies in DNA or even RNA, we might encounter into either structural perspective or even sequential perspective. And with today's current scenario, we can see that there are actually a lot of people involved in NGS, which is very much into the sequence. But the importance of structure, as specifically in protein, is that a structure is actually 3 to 10 times more conserved than sequence. And because of that, that is so important that in order to get a better understanding of their responses in the biological process, it's very important for us to understand the structure and also to get hold of the structure. So these are some of the common, input files that we see from a bioinformatics. I would assume that most of the participants here are bioinformatics background, or perhaps you perhaps are from biology background, but you wanted to get into bioinformatics because of various reasons. So these are some of the common input files or even output files that we see in order to let us perform our job. But what I wanted to bring you in is that a lot of the time, even as a biologist yourself, that when we talk about protein, this is perhaps something that you imagine, yeah, because protein for a lot of people, especially those working in the lab, is always in the form of powder. But for a lot of bioinformaticists or even researcher, we often see this instead of this. And as a structural person, I would personally look into this as well. Yeah, so if you ask me how useful it is to look at it this way or this way, I would say obviously this way. This is a three-dimensional way that allow us to have so good understanding about the particular structure that we're working on. And of course, as technology progress, we are also looking into this perspective. This is a 3D printing of a dengue virus. That is actually my hand, so you're seeing that. So that is actually the size of a dengue virus capsid. And of course, with the advancement, now we are moving into augmented reality and also virtual reality in understanding structural bioinformatics. So moving on, structural bioinformatics is no longer just a 1D or 2D thing, just using your eyes and view it either on your screen, but we're looking into a more personalized feeling whereby you can touch, you can feel, and perhaps you can perform even some of the work with your hand as well. So this is where the field has evolved from, I would say, about 20 years back until today. Things have changed tremendously. So I also wanted to talk about drug research. I mean, this is probably one of the reasons why we have this gathering today. We talk a lot about drug. So a lot of time, this is represented, I guess if you ask a lot of people who are in drug discovery work, they would tell you drug research is like the search for a needle in a haystack. It's totally fun, I would say, fun and challenging. So just to find that little tiny needle in that haystack could probably take you a very, very long time, and of course with a lot of money that is involved. So this is actually the process that you can see. For a traditional drug discovery way, it actually begins with just doing some target identification, target validation, lead identification, candidate optimization. Now, this entire process itself could take you up to about 10 years before you can even bring something quite promising or something quite solid into preclinical. Now, preclinical is the part whereby you perform some, say, like animal studies, or even perform it at a cellular level. So this is what we call preclinical. And of course, finally into the clinical stage, whereby we are actually experimenting with real human subjects, and this could take up to many years just to check on the effects. So this is in terms of the time, and in terms of this is in terms of the time. But if you look at in terms of the compound, how much compound we actually get involved. So this is actually again a very simplified model to show you that, you know, if you start off stage 1 doing drug discovery with only 10,000 compound, when we move to stage 2, ultimately we are only interested to look into about 250 compounds. I guess the reason is very simple. If you are doing it in the wet lab, you can't be bringing all 10,000 compound into the lab, because we are looking into the cost, we are looking into the time. You know, it's simply too much to waste, or if we can't be assured of the success rate, perhaps there should be a way for us to do this filtering process. And by the time it reach into the clinical stage, it should only be about 5 compounds. And these 5 compounds also eventually goes through the phase 0, phase 1, phase 2, phase 3, and ultimately phase 4. So even at the clinical development itself, there are already 5 phases that scientists have to go through. And of course, finally, that particular one compound that managed to make through into the market, whereby it has to get the approval from the regulatory board. FDA would be the common ones that we see. Well, even each and every country has their own regulatory board. For each and every drug to get approval, that actually takes a long time. So to sum up this whole thing, we could see that to develop one drug, to even make it into the market, or even to put it into our mouth for consumption, we need to spend approximately 1 billion US dollar, and take approximately 10 to 15 years. Now, this is a very, very long process, especially in a desired situation like what we are currently facing now at COVID-19. We certainly do not have 10 years to wait for COVID-19. And therefore, you know, a lot of people has been discussing whether this entire stage can be shortened, can be made into a very fast way, so that, you know, we can get at least some candidate to work on, especially now during this COVID-19 situation. All right, so to address that, in drug discovery, this is a traditional way of doing drug discovery, whereby I look into more computational perspective, because we are in bioinformatics. So this is very much into the work, pardon me, okay, yeah, computational work. So you can see that in terms of the drug discovery, there's actually 2 major field, you know, the structure based and also the ligand based. So for structure based, we often heard about ligand, docking, homology modeling, quantum mechanics calculation, molecular dynamics simulation. I believe these are some of the methods that you probably have heard, but maybe you're not very sure what it is. Please allow me to explain it in a while, in a very brief way. Hopefully, you know, you can get it understood. And of course, besides the structure based, we also have ligand based. So you probably heard of QSAR, pharmacophore, or even similarity searching. And finally, if we could combine them, hopefully we can better optimize the lead, and finally it can be a better drug candidate. So to sum up the whole thing, again, in today's context, we are not only looking at the traditional drug discovery work, because we need to be a little smarter, I would say, or more rational. So there are a lot of combination kind of methods that could be used in order to make this method to be effective, or hopefully to shorten the entire duration to discover a potential inhibitor or even drug. So like in today's context, we talk about NGS, there are a lot of human genome sequencing that's being done. So a lot of NGS work being done. And of course, from a proteomic person and also from a structural person, I will always ask my colleague and say, do you have anything interesting from the NGS that I can take and continue with doing drug discovery and all? So these are all very important questions that we need to ask our counterpart. Yeah, and also the development in terms of three-dimensional. So structural perspective, this is also about getting the data. How do we get effective and also good data? And of course, for most of us who are in structural bioinformatics, we are basically at this level. So a lot of work are actually being done at doing the database of active compounds, or even target and also binding site identification, or perhaps doing some docking, or also some virtual screen, or maybe some molecular dynamic simulation, and of course, finally get it tested in the lab. All right, so I talked very lengthy about structural bioinformatics and also drug discovery. So how do we connect them? Yeah, so we often use structural bioinformatics as a tool to help us to solve various problems. So in terms of in silico protein structure prediction, there are three main methods that it could be used to do structure prediction: homology modeling, fold recognition, ab initio, and of course, performing molecular docking studies, such as something like this. So if you can perform molecular docking simulation in an iterative way, and if you can perform it over 10,000, 100,000, or even million times, we actually name it virtual screening. So this kind of work of doing virtual screen actually allow us to understand or even study whether any of these chemical compound can be used to bind on a particular drug target, and whether it is useful for us to move on to test them in the lab. And of course, if you can see, there's also a different kind of structural work that can be done, because it's not just about getting it docked at a particular target site, drug target binding site, but we also wanted to know what happened after you bind them if we put them into a simulation and try to run it for a period of time, and what will happen then, whether the ligand will continue to stay at the binding site for it to have any informative or even what is the word, any any nice activity, or nothing happen. So it's very important as well to move into the simulation perspective to check how well they actually stay. So I will share with you a little bit of some of the work that me and also my students have performed in the past. So we actually focus, this work was done about 9 years back, that we actually focus on plant. So this plant Ganoderma lucidum was used to study on the potential of anti-HIV, and we had quite a nice quite some nice result to move on. So we actually performed this in computational perspective. And a few years later, we did something similar, not on Ganoderma lucidium, but on understanding toxoplasmosis, which is again a very, I would say, an NCD, and a lot of this kind of disease that has been neglected by a lot of us. So we wanted to study, in terms of computational, whether we can have meaningful insight on the particular drug target. And back then there was no drug as well. So for us, the objective was very clear, was to try to determine whether there's any potential inhibitor that we could develop from computational method. So few years back, I also work on this work, which is on simulation, where we study on antibiotic. So understanding antibiotic again is very important, because that actually changes how antibiotics should be taken. So this was a work done by me and also my team back then, that we were studying, and we simulated it using molecular dynamics simulation. And besides this work, we also did some calculation just to understand like how much all of these antibiotic actually stay in each and every of the target, and how are the effects like, in order to keep them in the binding site. Of course, the ultimate aim of doing this kind of structural bioinformatics work is actually to understand, and it's actually to develop drug as well. So this is a very good summary that I pull from this paper that talks about what kind of drug that has been successfully developed using computational way. So you can see some of these drugs that's already in the market, and these are the drug target and what are the disease that they actually target and what are the ways they actually use. And we are talking about computational way. So this is solely some of it is structural bioinformatics work. Some of it is more into ligand based. Although it's not really structural bioinformatics, but because it utilizes computational way, so somehow I think it's also wise to mention here as well. So the success rate of doing computational work and from computational all the way to getting the final drug is actually there. So it's not saying that, you know, that there is none. There are a number of examples which you can see now, and that shows that how important it is to use bioinformatics work to actually complement into doing this kind of drug discovery and ultimately being a very successful drug that is being targeted into the market. So next, I also wanted to talk about how the big data that we are currently facing now and how do we relate them with bioinformatics. So this was an article published by Caltech on about big data. How big data has actually influenced computational biology or even structural biology in shaping the future of our health, and also there are many other different perspectives that we can look at. So you can see this was a paper published in many years back that talk about the challenges of big data. And although big data is something that we talk about, you know, very frequently for probably for the last 3-4 years, but actually big data in biology already happened about 9 years back. That was because back then there was already a data explosion. Well, this kind of data explosion actually happens because of genetic sequencing. So this was quite an old data, I would say. This was in 2012, that was 8 years back. So you can see in terms of the data pile, the data pile is actually just 1/10 the size of the data storage at CERN. So if you know about CERN, CERN is actually the Europe Particle Physics Laboratory near to Geneva, Switzerland. So this data pile is actually only 1/10. So you imagine every year in CERN, they actually produce about 15 petabytes of data, and that is also equivalent to like what we are seeing currently in the biology perspective. So the European Bioinformatics Institute currently stores about 20 petabytes of data about genes, proteins, and so small molecules. And this genomic data is about 2 petabytes per year. All right, so you can see from this perspective, is projected in 2025 that genomic data is going to exceed even whatever data that we see, Twitter, YouTube, or even astronomy data. And just to give you a context, how much is petabytes? You can see from this how many zeros here. So a data scale here will help you to imagine what kind of data that we are talking about. So we are talking about 10 to the power of 15 kind of bytes of data that we see on a yearly basis. And there's also a dramatic decrease in terms of time and also cost to sequence data. So you can see that in order to sequence data, it was 100 million in 2001. And in 2016, it was only $1,000. And today, it actually drops to about 10. So you imagine the cost to actually perform this kind of genomic information is actually so small nowadays. And this is, again, the same information that we are looking at. So if you can see again, in terms of real time, I actually took this data on 15 December, about 5 days ago, from GenBank. You can see how much data that we are actually producing. And using this data, we can actually see that, you know, there's so much potential that we can do with this data. And mind you that this data is actually available free of charge. You don't even need to pay a single cent to get this data, and you can just get it download, yeah, to your own to your own PC. So again, perspective wise, this is on the GenBank. But if we look at the structural data as of yesterday, it was only 172,000 structures. And you can see this number is actually very, very small compared to the gene data that you get from the GenBank. And on top of this complexity of not much data that we get from 3D structure, the good quality structure, because data has to be good quality. And just when I started my talk, I actually talk about how important it is to get good quality data. And you can see that good quality data that falls into this region is only this, and it is very, very small. So these are usually some of the I would say the usable data that we can do in order to perform the drug discovery work. Yeah. So this number is actually so small that even we felt that although we are in structural bioinformatics, but because of the complexity of structural information and not able to get a good data source, so there are different ways. There's definitely a need to improve on this particular way. So if you can see that, AI has come into the picture in structural bioinformatics work, where you can see artificial intelligence or AI. Basically, the definition of AI is to mimic human behavior by simulating human intelligence using computer methods. So if I sum it out in AI, there's actually four main approaches that we can see from AI perspective: from the machine learning perspective, the deep learning, natural language processing, and also finally robotics and IoT. Although four of them sounded the same, but actually they are totally different in AI, where you can see machine learning very much focused on the input data that we get, and we repeatedly optimize it using the data we generate. That's called machine learning. While deep learning is very much into getting the knowledge out of it by simulating the neural network. So this is a deeper kind of learning curve for that particular system that you have. And of course, some of us are also involved in NLP, natural language processing, whereby this is more into automated speech recognition. Yeah, and also we look into robotic and also IoT. So this is more into collecting the data that we need from the right hardware that we required. So this is into the robotics and also the IoT. So how do we get all this AI into drug discovery? So we have heard about drug discovery, heard about AI. Now let's try to combine them together. Although it sounded a bit challenging, like how we can do that, but actually, there are a lot of ways and a lot of effort in putting AI into drug discovery. So if you remember, I vividly explained about this just now when I started my talk. And let's see how we can actually combine machine learning into this pipeline. So there are a number of generative model that we can fit into machine learning, including reinforcement kind of learning at this level, at the screening level. And once we pass the screening level, when we go into the filtering level, this is where we can put into deep learning method. And finally, we can do some graph embedding to make it to make the system learn better. So not only combining this, but you can actually see that there are actually a number of studies being done, like how much what is the need of combining AI into drug discovery. And you can see that 36% of global AI in drug discovery market was actually done by deep learning. So this is actually to show the success rate of doing that. And if we want to see what kind of who are the main stakeholders in putting this AI together with drug discovery? Of course, can't forget, pharmaceutical companies. They are the major stakeholders into doing this. And of course, we have there are also AI introduced into it. And of course, there are some technology company as well. So you can see these are all the three main field that comes together in order to bring AI into healthcare and also drug discovery. And this is another perspective of how where are they involved specifically at each and every stage. So even started from the target discovery itself. So you must bear in mind that for some of us that is doing structural bioinformatics, we are very much involved at this level, the preclinical level. So preclinical level, of course, is the part where we use a lot of AI to help us to speed up our entire discovery process. And if you can see this as well, these are some of the example of the AI companies that is being active currently in doing drug life cycle as well. So these are the big companies, how much they have invested into producing a good drug so that it can be get into the market. And these are just some of the snippets that I managed to pull through from some very important literature about AI in drug discovery. So there's one here, one here, another one here. Of course, they're studying at different perspective as well. Not only studying at some of them study at drug discovery, but some actually study even at the biology system itself. So if we conclude all of these things, you can see that what are the challenges that we have. So the first challenge that I can see is that it's a data challenge. Why do I say data challenge? Because there's obviously a lack of high resolution 3D data. Just as how I've explained, the data that we have received from PDB is probably only about 10% of it compared to the genomic data. And I will say and out of those data, only about 10% of it is really functional. And what do I mean by that? I'm talking about in terms of the quality of the data. So it's also, again, important to look into the resolution of the data and how much we can trust the data. Second challenge, I will probably put into technological perspective. There's obviously a need to improve techniques and also method. Mind you that some of these method actually is as old as probably 50 years old. And I'm not saying that they are not good, but it's obviously need to have a little tweak here and there to improve. And perhaps with the AI method that I've just introduced to you, perhaps it's actually time that, we start to combine AI method with some traditional a traditional knowledge base, and maybe we can enhance that with a new algorithm and hopefully the entire simulation or the screening process will be done even in a more accurate way. And another important problem that we're actually facing is actually storage. Today we're talking about petabyte, and let me ask you, how many of our lab actually equipped with this hardware and also the cost to help us to store this data? So this is also another challenge that we face. And most importantly, I would say in terms of the validation. So it's not just about doing this in the computer lab, validating against with the database, but there's also a need to validate with experiment and also clinical studies. And obviously, we need to work together with other fields. We can't do this all by ourselves. There's always a need to do collaborative work with other labs and also other fields. So if I were to sum up everything, there's this 5 main key consideration when we wanted to adapt this AI solution. First, it has to improve the access to robust, reliable data. Secondly, it must be able to recognize the destructive potential of tech giants. Third is to increase the diversification of drug discovery pipeline. We're not just looking into the traditional pipeline, but we are looking into a more creative way. And of course, acquiring skill. And when we talk about acquiring skill, we actually talk about the scientist, the researcher. Are researchers actually ready to do this? And of course, finally, the critical thinking part and also the performance indicator. So I actually echo with what Dr. Adais has shared before this. Doing bioinformatics is not just about skills, soft skill. Sorry, it's not just about hard skills where you move into the programming part, the analytics part, but it's also a lot about that mind, that critical mind. And I'm going to say that a lot of us are probably not trained, to think critically. That is why it's so important to have a team to brainstorm together. So before I end my talk, I just want to bring up this topic again, this question. In fact, this is the title of my talk. So my question is very simple. In today's context, is structural bioinformatics still relevant? Now, my simple answer is yes, it's a yes. It's not just a yes, but also improve. So there is importance. It's so important to improve on the method that we have, not only doing the traditional structural bioinformatics way where we used to or we are comfortable in doing, but it's perhaps time that we have to look into using AI or even help from different kind of technology to help in that perspective. So this is actually my contact information if you're interested to find out about our team, our group in Perdana University, we do have a bioinformatics team in Perdana University, which we not only work in structure, but we also touch on various perspectives of bioinformatics. So you can get in touch with us. Please like our Facebook, our Instagram, our YouTube channel as well. And if you have any question, feel free to ask here. So with that, I'm going to end my talk here. Thank you very much for your attention.
Madam, yeah.
Yes.
I'm Professor Kripanidhi.
Hi, Professor.
Yeah, we wish to know what are the AIs, because we are basically with the biology background, biotechnology background. You have been stressing much on AI skills.
Sure.
So may I know a few?
Okay, so if I could,
which are useful in structural bioinformatics.
Yeah, so if I could explain to you, Professor, we should always start with the basic, which is the structural bioinformatics. So there are a lot of perspective that we need to understand the structure-wise, the protein structure, and also what are the methods that we can use. So probably some of the methods you have heard, like docking simulation, ligand-based drug discovery, like QSAR, molecular dynamic simulation. If we start with all these methods, and then whatever method that we can use to improve on that, those would be an additional on the artificial intelligence method. So Professor, maybe you can start with the structural work first. Then on top of that, we can use all the artificial intelligence method to help us to I would say, fine tune it to make it more presentable, and hope that we can filter the result into a better way. I hope that I answered your question.
Yeah, that's true, madam. So is there any possibility for our students to come over to you for internship, madam?
Oh, sure, definitely. Something that I forgot to share is that in Perdana University, we actually accept interns. So we accept a lot of intern students into our university to do their internship. But because of the current lockdown, most of them couldn't come into the country, but we actually accept virtual internship. And the school also perform a lot of training, not only on structural perspective, but also in bioinformatics training. So Professor, if you're interested, you can get in touch with me, or even go to our Perdana University website to have a look. What I can do is that I can drop my email address in the chat box. So if you have any question, perhaps we can communicate from there. Thank you.
Thank you very much, ma'am. It was really an insightful lecture.
Thank you.
After such an emphasis on AI and structural bioinformatics, we really feel honored to have you, have your esteemed presence amongst us.